right, this is part 2 of your midterm review. We'll be looking at questions 10 to 18. So question number 10, what is a polypeptide? It's just a long chain of amino acids. And remember, all those amino acids come together and they're going to form proteins. So just a simple definition question there. Number 11, what property of water makes it a universal solvent? Well, we have to know what a solvent is first. And a solvent is something that dissolves other substances. So it being a polar molecule makes it a universal solvent. And when we talk about a polar molecule, that means where one side's positively charged and the other side is negatively charged. Number 12, which of the 4 macromolecules is insoluble in water? Well, you got to know that there are 4 macromolecules, and we'll cover those on the next slide. But there's one that is insoluble in water, and that's going to be lipids. Now, remember, the cell membrane has a hydrophobic tail, hydro meaning water, phobic meaning fearing or hating, and the hydrophobic tail will not mix with water. Think of it like this: when you make desserts or something at your house, can you wash off oil with just water? No, you have to use soap, which will break down the lipids, making it easier to wash off. All right, number 13, know all 4 types of macromolecules, their structure and components. So the first one is carbohydrates, and that's going to contain carbon, hydrogen, and oxygen. It's always going to be in the shape of a ring or a circle, and that's going to give us our quick energy. That is energy for short-term usage. Then we have lipids, which also contain carbon, hydrogen, and oxygen. Well, if it contains the same thing as a carbohydrate, how can we even tell the difference? Well, we look at the shape, and usually it's going to be in the shape of a straight line. And then we have proteins, which contain carbon, hydrogen, and oxygen, but it has one more thing, and that is nitrogen. So it has the same 3 elements that lipids and carbohydrates do, but it only has one extra item, which is nitrogen. And that brings us to the last point, nucleic acids. So it contains all 4 of the previous ones, carbon, hydrogen, oxygen, and nitrogen, but the one additional thing it has is a phosphate group, and that is nucleic acids are your DNA and RNA. All right, adhesion versus cohesion. Very, very simple. Scientists have done us a favor with co meaning together. So let's start with adhesion, then we'll talk about the easy one, cohesion. So adhesion is when 2 different types of substances cling together, and the key part is 2 different types of substances. They're not the same substance. Cohesion is the attraction of 2 or more of the same types of molecules, like water droplets on a windshield. When you're driving down the road and it's raining, or you're watching rain out the window, you'll see that water sometimes begins, or not sometimes, it starts to pull together, and that's because it's attracted to itself, and that is cohesion. All right, moving right along. Compare and contrast DNA and RNA. So it says compare, so we got to find out how they're similar. It says contrast, we got to talk about how they're different. And remember, all of this should be a review. If you're using this video to learn something for the first time, you're not going to get as much out of it. You really need to have a foundation. We're just doing this to simply review. So all of this you should have heard before, because I know we've gone over it in class. So comparing DNA and RNA, well, they both have adenine, cytosine, and guanine. That's the A, C, and G. Both of them are nucleic acids. For the most part, that's how they're similar. So how are they different? Well, DNA has thymine, which is the T. So they have DNA has A, C, G, and T. RNA has ACG, but instead of thymine, RNA has U, which is uracil. They're also different in the number of strands they have. DNA has 2 strands, while RNA has 1 strand. One thing that is not listed here is also the sugar. DNA is deoxyribose. That's why it's called deoxyribose nucleic acid. So the D is for deoxyribose. And then the sugar in RNA is simply ribose. That's why it's called RNA ribonucleic acid. So the sugar in DNA, deoxyribose. The sugar in RNA is ribose, which is not listed here. So you might want to add that to your study guide. What effect do most mutations have on an organism? Well, mutations are random and they happen all the time. So what effect do most mutations have? Most mutations have no effect on an organism at all. There's a great myth out there that talks about mutations often kill organisms. Even though that can happen, Most mutations have no effect. All right, number 17, transcription versus translation. Okay, so let's talk about transcription, which comes first. If you're trying to remember which one comes first, just put it in alphabetical order. Transcription comes before translation, so that will help, and transcription will always come first, and that's when mRNA is going to make a copy of the DNA. So transcription and translation is part of protein synthesis, how proteins are being made, and the mRNA has to come into the nucleus and make a copy of the DNA. The DNA is too large to leave the nucleus, and we looked at that when we extracted DNA in our extracting DNA lab. So the mRNA will come in, it will make a copy of the DNA, and that will lead to translation, and that's when mRNA is paired with tRNA. And when we talk about pairing with tRNA, we're talking about those bases being complementary, A pairing with U, C pairing with G, G pairing with C, and U pairing with A, and we'll get more into that shortly. How many nucleotides are needed for a codon? 2 codons, 3 codons. So here we have this word codon, and we need to know what that means. Well, 3 bases, which are the nucleotides, are needed for 1 codon, and codons are found in mRNA. Anticodons are found in tRNA. Either way, you still need 3 bases. So if we have 2 codons, 2 times 3 is 6, we would need 6 bases. For 3 codons, we would need 9 bases. If I had 21 bases, how many codons would that be? Well, 21 codons divided by 3, that would be, if we had 21 bases and there's 3 bases that make up a codon, 21 divided by 3 would be 7. So that would be that. So if you know how many total codons you have, you're going to multiply by 3. If you have the total number of bases, you're going to divide by 3. So for example, I have 9 bases. How many codons is that? 9 divided by 3. Here I know the amount of codons, so I'm going to multiply by 3. 2 times 3 is 6. Same thing here. Here I'm given the amount of bases, so I need to divide. 3 divided by 3 is 1. So 1 codon equals 3 bases. Okay, that's going to be it for this one. If you have any questions, please let me know. We'll see you guys in part 3.